dance by the water neath the Mexican sky. Drink some margaritas by a string of blue lights. Listen to the mariachi play at midnight. Are you with me? Are you with me?
Well, once I have done a little bit of coaching, now I would like to transfer the microphone to Rafa, who has proposed Guido as the speaker, invited speaker for today. And this is yours.
Oh, thank you. So, first of all, thank you because today we have many people here, so thank you for coming. So I met Guido 2 years ago in a conference in Prague, and actually we met having dinner with other people, with other researchers, and then after that I went to his conference and I really like it because I think it's a very good example how to translate fundamental science to the field. I think for the institute that is a very good example to follow because we have so many possibilities here. So Guido is professor and chair of translational plant biology in Utrecht University. Basically, as I said, he's focused on translate fundamental science to applied by private-public partnerships. So Guido, is a great honor to have you here today, so the floor is yours.
Thank you very much. Is it working? Yeah.
Probably you need something, some help? Or
Yeah, if you can start.
you minimize this. And now you, okay.
Here we are. Okay, thank you very much, Rafa and Javier, for the nice introduction and for having me here in Malaga. A bit lack of sunshine, but then I can justify that I go back tomorrow already. So, but and also good, Javier, that you reminded us that we only have one planet to live on, so we should take care of that planet. So I think a lot of our work is also on sustainability and environmentally friendly agriculture. So I think that illustrates that also very much. So indeed, today I would like to tell you more about public-private partnerships, where we try to translate fundamental plant science to crop improvement. And I will give 2 examples of large projects that I've been working on. The Let Us Know project, of course, on lettuce, and Crop XR, where we work on multiple crops. So I would first like to tell you a bit more about working in public-private partnerships, then tell you about this Let Us Know program, which was really aimed at generating insights into plant architecture and resilience using a lettuce diversity panel. And then in the last part, I will talk about Crop XR that started 2 years ago, 2.5 years ago, like a virtual institute focusing on smart breeding methodology for crop resilience. So translational research and translational plant biology is also the name of the chair group that I'm leading in Utrecht. This research is needed to bring fundamental science to an application. And what I always like to stress is that you can only do good translational research if you have good fundamental research, because if you don't have innovative ideas, there's nothing to translate. So it's about going from the lab to the field, and I think in your institute you have all these possibilities to bring research from the lab to the field, and you also know that this comes with many challenges. And in the group in Utrecht, the Translational Plant Biology group, we focus on 2 topics in relation to plant development and on disease and immunity. And the focus, the way we execute most of this translational research is through public-private partnerships with the Dutch plant breeding industry, and we are fortunate to have a lot of companies in the Netherlands that are really advanced in breeding of vegetables, potato and ornamentals. So this collaboration between academics and industry is of course really important, and by nature, the scientists, the researchers, they're very curiosity driven, and they're not always thinking about the applications or how to, what it means for real life problems. But when you are collaborating with industry researchers or with farmers, you get better insights into the problems that they encounter, and that helps you to think about this translation, of course. So it's really about hearing about the problems and also realizing the importance of certain traits and what they could mean for crop improvement. And this translational step is then needed to really make use of these scientific discoveries. So I thought it would be good to tell you how I got into this public-private partnership. So a long time ago, in the previous millennium, I started in Utrecht to start my research group, and I started working on a fundamental model system, Arabidopsis, and the downy mildew pathogen, because I wanted to understand what now makes plants susceptible to disease. At that time was really very good timing to work on Arabidopsis because the genome was just published in 2000. And then in 2002, I got my first public-private partnership with 2 vegetable breeding companies to work on the genomics of downy mildew, because at that time we didn't have genome sequences available yet. Also, through other parts of my research, we cloned a number of susceptibility genes in Arabidopsis, DMR1 and DMR6 for downy mildew resistant. And one of the companies that I worked already with was interested to develop that further and see if we could implement that in crops. And since 2007 until 2 years ago or so, so for almost 15 years, we collaborated intensively with Enzazade on the use of this DMR technology. This was patented by us, and they worked on realizing that in crops. I will not talk about that today, but if you're interested, I'm happy to discuss the use of these susceptibility genes in disease resistance breeding. Then since 2007, we started working also on crops, because downy mildew was also the most important pathogen in vegetable crops like lettuce and spinach. So this downy mildew we studied very intensively together also with companies. We had a large national program called Center for Biosystems Genomics. So there multiple companies were collaborating with multiple universities. And also at that time we had a partnership with one of the companies on disease imaging. Maybe the last 2 are most important. So 7 years ago we started this Let Us Know program with 7 companies, and now I'm also leading this CropXR I will tell you a bit more about, with more than 20 companies in the Netherlands. Okay, let me move to the first public-private partnership, this Let Us Know program, which is really meant to provide insight into the biology of resilience and architecture using multi-omics approaches. And the nice thing about the Let Us Know program is that we use a diversity panel of 500 lactuca lines. I will tell you a bit more in a minute. Actually, no, I don't have that slide, but these 500 lines, they are 200 Lactuca sativa, so cultivated lettuce, 200 Ceriolas, so that's the closest wild relative, and 50 Virosa and 50 Saligna lines. And they represent a very nice diversity panel. So in this program we work with 4 universities and 7 vegetable breeding companies. And as you can see on the top, we work on architecture and resilience, but a big part of the program was also on the data side, where we invested also in bioinformatics and machine learning approaches, and in the center technology, which means also transformation, genome editing, etc. So one slide also on working with these vegetable breeding companies, and it's good to know that these breeding companies are quite technology driven, so they are also R&D intensive. A number of these companies are family owned Dutch companies, and they really invest a lot of their revenues back into research, almost 30%, which is really very high. If you compare that to electronic sector, for instance, this is really high. Also, company researchers, they really understand the importance of the science and also of the fundamental research. And many of our graduates find jobs in these companies. So also many of the people that we work with in the company, they have graduated at one of these universities, done their PhDs, etc. So they also know what it takes to do research. And the question that I often get is, how do you manage to get to work together with companies that are competitors? Well, the answer is that you should do research in what we call the pre-competitive space. So that means also more towards the fundamental science. Because when it comes too close to breeding, they are not willing to collaborate because they are developing their own varieties and everything. So as long as it's strategic, pre-competitive, then it's good. So it provides, such projects provides them with new knowledge and tools, methodologies, human resources. Also those PhDs, they find jobs in the industry. And industry can also provide materials. They grow lettuce lines for us. They provide seeds. They make crosses, F2 populations, etc. Field trials also. Okay, I had this slide with different lactuca lines, and as you can see from the phenotypes of these plants, they are not only genetically very diverse, but also from their phenotypes, of course. And the nice thing is that with all these people in the program, we are working on the same set, so we can compare all our data because it's always linked to the same genetic set. Well, we are looking at different biological problems in lettuce, such as early bolting and flowering, which is really also environmentally controlled. So also heat stress is a big inducer of early flowering and bolting, and then lettuce becomes bitter also. Tip burn is really a very serious problem. It's a physiological disorder. Salinity stress is important, also in Spain, I'm quite sure. And then my favorite is, of course, the pathogen, downy mildew, as an important topic. And as I said, in this program, we really learned also how to work with the gigantic amounts of data, phenotyping data, RNA expression data, genomic data. We were prepared a bit because we had a lot of computational scientists in our program, but still it was not enough actually. So one part is on the genome side. I have a few examples from our program, from results also. So Dirk-Jan van Workum, one of the PhDs in the program, he is working on pangenomics. So he used available genome sequences for all these 500 lines. We have genome sequences available to build a super pangenome. And super pangenome means a pangenome is within a species, and super pangenome means multiple species because we have sativa, cereola, saligna, and virosa. So then you can also see what is shared between species or what is lineage specific, and that provided a lot of information, especially that there were a lot of presence-absence polymorphisms and also copy number variation. That's really important. He published that, and now Dirk-Jan is also, well, he's defending his thesis, but he also worked closely together with Richard Mitchamore at UC Davis, where now very high quality genome sequences of multiple lines are being produced, so we get really very high quality pangenomes now. So that's indeed what he's working on, and then you can also zoom into your region of interest, and I will give an example later. So, as I said, we are collecting lots of data, especially on the 200 sativas and the 200 cereolas. Phenotyping in a field trial experiment, in growth chamber experiment, and in all kind of lab-based assays. We've also done extensive expression analysis. I will come back to that in a minute. We almost made 6,000 RNA-seq profiles from lettuce. So this was a gigantic resource. And the thing that we can do now with this diversity panel is use that for genome-wide association studies. And we've developed multiple flavors of these GWAS analysis. The normal SNP, the polymorphism-based GWAS, but also k-mer-based GWAS and the presence-absence polymorphism GWAS. And very often we find the same peaks, but still we find differences between those approaches. Then also we are using more advanced methods such as multi-omics analysis, and we are now getting into AI approaches as well. So I put this movie in for you, Rafa, for our field trial that we did in 2021, where we had duplicate plots of the 200 sativas and the 200 cereolas, and we were monitoring them by visual scoring them, but also sacrificing them. And there you see the field robot from our phenotyping center going over the field with hyperspectral and RGB imaging, collecting a lot of data. Also the drone that is used here to make this movie is also used to measure height and color, etc. So that also has been an enormous data resource, and it has taken us quite a while until we processed all that data and were able to use it. So we're still digging into the hyperspectral data now. So one thing that we found from the field trial, and this is just an example, and this is on the bolting, the early flowering, and there you see this is not a Christmas tree, this is a lettuce variety. And clearly from all the measurements that you see on the right-hand side, these are all different measurements at different days or different features. Most of them mapped back to a position on chromosome 7 that contains the phi C locus. And clearly there is in the lines that are, the polymorphism is a truncated phi C protein. And interestingly, the same position was also recently found by colleagues in Wageningen to be involved in the circadian clock. And this was published in New Phytologist last year. So our group also contributed to this paper where both bolting and the clock are very much affected by this phi C mutation. And apparently this has been selected for very early on in the domestication of lettuce to make sure that it is late flowering. Yeah? So this lines that have this mutation, the clock is also not around 24 hours, but more around 27 hours. So, and that disturbance leads to delayed flowering. If you like, you can read more about it in this paper. So I said we also used the drone, and we recently published that first paper on the phenotyping of the field trial, where the drone was used to do all measurements and then use that for QTL analysis or GWAS analysis. So actually what we first discovered by going into the field doing all these different measurements, the drone also did for us, because it could measure the height of the plant, and the height of the plant says something about the flowering. So clearly, and those are the lower two graphs, you clearly see the phi C locus popping up there again, and this is related to height and also even red edge on the plants. But and on the top it's the green-blue ratio, so that's where more with anthocyanin accumulation in the plants, where we also find really good peaks that we can associate with biosynthetic genes. So take-home message is the drone can save you a lot of time for these kind of measurements, but you have to learn how to work with that type of data. So then one example from my group where we worked in the Let Us Know program on pattern-triggered immunity, and this is the immune response that are triggered by molecules that are produced by pathogens. And we exposed our 200 Lactica sativa lines to different PAMs So these are heat-killed bacteria, flagellin 22, which is a part of the flagellin protein, chitin, and NLP24. And NLP24 is a small peptide, 24 amino acids, synthetic peptide, derived from a protein that is produced by dinosaur mildews. And in Arabidopsis, we know the receptor. But in lettuce, it seems there is also recognition, but lettuce does not have this receptor. Not the RLP23, it has a different receptor probably. So, and as you see on the right, on the y-axis there are the 200 different lines, and then on the x-axis you see the ROS burst. And the more red or yellow, the more strong the ROS burst. And that's a signal of recognition and the immune response. And clearly for NLP24, this is perfect for a GWAS. Half of the lines are blind to the peptide, they don't respond at all, and the other half show a very strong response. So in the GWAS, this gave an enormous peak on chromosome 4, and then we went to look for the candidate gene. And luckily, at that time, Richard Mitchelmore in UC Davis was already working on these high-quality genome assemblies, and they were very helpful, because the reference genome, Salinas, that line does not recognize the peptide, so it does not contain the gene that we were looking for. But luckily there was another cultivar, Ninja, which was responding to the peptide, and we found one single gene, LNR, we named it lettuce NLP receptor, that is present in all the lines that respond, but absent in all the lines that are blind to the peptide. At that position there is a transposon. So this was really nice. Actually, we were first focusing on this region here. You also see a lot of blue stripes. That's a whole cluster of receptor genes. So it was very likely that our receptor would be one of them, but actually it was next door. And then, so without these high-quality genome assemblies, we might still be looking for this. So it's really, the value of this is really not to be underestimated. So we cloned this candidate receptor, and we were able to show that it is really the receptor by transforming it into this Salinas, which is not responsive. And then if you look in the control, the empty vector control, you see that it's not responding to the peptide, but it is responding to flag 22, another peptide inducer. But when we transform in LNR or a GFP fusion with LNR, we see that the plant regains the capacity to recognize the peptide. So this is a receptor-like kinase, and here you see the extracellular domain. So the receptor sits in the membrane, it's monitoring the environment, and in red you see the NLP24 peptide. This is an AlphaFold model, but the peptide is strongly binding to the receptor, and we have also identified the most crucial positions, and when we mutate those, we also lose recognition. So this is really we are quite sure that this is the receptor, and we are now further studying this receptor and also its potential application by introducing this receptor into other plant species. So we have introduced it now in potato and are also looking into other crops to see if by adding this receptor, it is a GMO approach then, so probably not for Europe at the moment, but you could think of field crops where maybe such a receptor could improve the immune system of the plant, because NLP proteins are produced by many different pathogens, oomycetes, fungi, bacteria, not all of them, but many produce NLP proteins. Good, then within the program we also, as I said, we produced a lot of gene expression data. So we had on 400 lines, we have done 9 analyses, a shoot response, a root response to salt, far-red light and a control, and on leaves an immune response in response to salicylic acid, which is an important defense hormone, a mix of patterns and a mock treatment. So all these 9 treatments were done on 400 lines, so that's in and then with controls it's almost 4,000 lines. And then also we added another 2,000 samples for our compendium or an atlas, And this was mostly on selected reference lines, and we did time courses of treatments like immunity or light or salt, and also the different tissues. And here you see an example on the right where seedlings were dissected in maybe 10 parts, and they were all sampled also in time, and then we can now plot the expression of every gene you're interested in on these seedlings. And this, we just submitted the paper. You can also find it on bioRxiv, and this resource will also be openly available if you're working on lettuce, that you can see where your gene is expressed in different tissues. So these this expression data that we have done on the 400 lines, actually we are using that now for eGWAS, so expression GWAS. So we use the expression of every gene as a quantitative trait, and then you can map which loci on the genome influence the expression of each and every gene in the genome. And this is very helpful to reconstruct gene regulatory networks and also find how regulators influence the expression of genes. I only have an overview data here where, for instance, in the anthocyanin pathway, we could use this very strongly because we have also very nice phenotype data, we have the expression data, we can find the orthologs of biosynthetic pathways between lettuce and Arabidopsis, and this has helped us to focus on the important genes. And we did observe differences there where, especially at the regulation level, where different regulators are involved in control of the anthocyanin pathway. So this really helps you to reconstruct major regulatory mechanisms, and also stressing then that it's not always like in Arabidopsis, but it's very useful to use the information for Arabidopsis, but then it really helps you to find where is it different. So to conclude this part, so in this Let Us Know program, and this is a picture taken from one of our user meetings. We had meetings twice a year, a 2-day meeting where company and academic researchers would meet. Even at the plant breeding company, we had 3 meetings at the plant breeding company. This was at Rijkswaan. This was really working out really nicely as a kind of a team science. So the good experience from having led the Let Us Know program also allowed me to build an even larger program called CropXR, and I would like to tell you about what we're doing in that program. And this goes back to also the planet on the first slides, I think. We are exposed to climate change. We notice also with the weather here and the warm ocean, the water that is coming this way. We need to reduce pesticide use and other inputs in agriculture. And if we don't act in the no action scenario, then yield and quality of our production will go down. So there's smart interventions needed, and you can do that by improving your agricultural practices or cultivation systems. But in CropXR, we are really focusing on breeding extra resilient crops. That's what the XR stands for. It's not Extinction Rebellion, it's extra resilient crops. And it's really about the methodology also, and I will tell you in a minute. You can also think of the combination of the two. If you have more sustainable cropping systems, such as row cropping or mixed cropping or whatever, you might need to breed also specifically for those systems. So we're also paying a bit of attention to that aspect. And then resilience, of course, is, we define it as the intrinsic capacity of plants to effectively withstand and recover from adverse conditions and disease threats. So this is quite a broad feature, of course. We were able to build this initiative from 2 major funding schemes. So we were actually preparing already an idea for a larger program in the Netherlands. We had no idea how we would finance that, but while we were setting up that program, funding opportunities came along. So from our Dutch Research Council, there was a new program launched, a long-term program, so 10-year programs, and we were successful in the first round in the Netherlands. So it was on all parts of science, and 2 projects got funded, one on AI and one on plant biology. So that was quite remarkable. We had to compete with medical sector and everything, so. And then the other half of our funding we got from another initiative that was launched 5 years ago, the National Growth Fund. At that time we had a smart government that wanted to invest in innovation, yeah, and they prepared this big fund, 20 billion euros, and we got funding from that as well. When our new government came in, and we were halfway this National Growth Fund, our budget was safe luckily, but then they said, okay, we stop now, we spent 13 billion, we have 7 billion left, let's use that to reduce taxes on gas. Yes, okay. We now have a new government in place, starting next week, I hope, and they have, again, attention for innovation, education. So I hope they stay for a bit. So the good thing is that we have budget for a 10-year program, so we could also develop a long-term vision. It's not 4 years and then it's ending and you're not sure. So, and yeah, to get to crop resilience is a big effort, so it's good that we have time for that. So besides the universities here on the right-hand side, Utrecht, Delft, Amsterdam and Wageningen, and the funders, we have a lot of companies on board. Also Holland Innovative Potato, this is a consortium of about 12 companies in the potato sector. We have vegetable breeding companies, ornamental companies and 2 technology companies on board. And also we have an education program in which we collaborate with 4 universities of applied science that are also working in the green sector. Yeah? And then, yeah, this is the education program. We also have a serious investment in data infrastructure, but most of it is really going into the research of CropXR. And then we have this AgroXR, that is really the link to agronomy, so breeding for resilient cropping systems is in there. Yeah, so we work on different stresses, mostly elevated temperature, drought, and diseases and pests, and also the combination of these stresses. And although we are working with 20 companies, in the first 5 years, most of our research is still in Arabidopsis. Yeah, they're okay with that. But we are also working on crops, and this is especially in the second phase of 5 years, which will start in 2.5 years from now. The only exception is potato. At the moment, there is already a potato program running in CropXR. And I will tell you what we are doing in C1, but which is developing of the methodology, but we are studying different aspects of different physiological challenges, so immunity at elevated temperature, root development under drought stress, trade-offs, reproduction and temperature. We look at storage organs, that's C6 is with potato and onion. We have a social sciences program, and we have a data-driven program. And maybe on this slide I can explain best what we try to achieve in this program. So some 20 years ago, the genomics revolution started, and people started working with DNA technology, in plant breeding also. So that was really a big technology leap, and if you now go to these breeding companies, yeah, they have robots and it's all automated, and they do millions of genotyping assays, but it's really very much in-gene markers. They can follow a lot of genes, but they work still on relatively simple traits. So the challenge is now, how do we work with more complex traits? And if you think about crop resilience, that's typically a complex trait, and it's not a single trait. But the way forward there, we think, to achieve this technology leap is the adoption of modeling approaches in combination with artificial intelligence. And this is especially important because we're also living in an age where you can produce a lot of data, and we make only use of a limited amount of the information that is in the data. Yeah? So how can we mine that data even better? And I've already given you an example from our Let Us Know program, where we used the field robot and the drone. These are parts of the Netherlands Plant Ecophenotyping Center, which is a joint program from Wageningen University and Utrecht University. The units on the left here are in Wageningen, and on the right-hand side, these units are in Utrecht. And together they form this NPEC center. And as you can imagine, all these optical sensors and other measurements, they produce tons of data. And then in combination with RNA expression, proteomics, metabolomics data, we have huge data sources. And then we have this scientist that is overwhelmed by the data and how to use that. So we want to go from data to understanding to then inform breeding also. So here you see the massive amounts of data, of course, and we are especially working here at point 2 is how do we channel our data into models of processes. So we'd like to understand, so not black box artificial intelligence, but we like to understand what is going on. So we use mechanistic models to then use the big data sets to inform these models and understand how certain traits work, and also which knobs to turn to influence or improve those. And then at point 3, of course, a very important part is also how can you translate what we develop initially in Arabidopsis also to other plant species, and that's also a major challenge, of course. And the reason why we work with Arabidopsis is because most knowledge is there, so we can build initial models already of biological processes, learn how we can use data to improve our models, and once we know that, we want to use that in crops. So this is the important C1 project, the demonstrator, where we want to integrate those data sets and models. So we are doing experiments. We've just finished a big stress matrix experiment where plants have been phenotyped. This is all about seedling establishment, so the early phase of plant life under drought and elevated temperature. We have known data, and this provides us with initial models. And then the big trick is in combining these initial models with mechanical formulas, these models to understand how processes work, and that can be done also in a tissue-specific way. For instance, you can make a model in the shoot, in the hypocotyl and the root, also have communication between these models. So this is being set up in what we call the demonstrator project. And just to give an example on elevated temperature and drought, and this is a very simplified figure where we work with just a few important components that are known from literature that are important in drought and elevated temperature responses with SPA1 and COP1, Hi5 and PIF4. And we can describe levels of proteins by different mathematical formulas that involve expression, protein degradation, phosphorylation related to activation, etc., to then produce outputs on how the plant would grow under these conditions. And we are now combining this with the big data sets. So these are machine learning models that we generate from the modules that we generate from gene expression data and other data that we try to incorporate into these mechanistic models. So these are these differential equations. And if you're interested, you can read about that in this paper that was published last year, Badadan. It's how gene expression data is being used to create these modules to use transcription factor binding site data and other data, also using the big expression compendium that is available for Arabidopsis to then make differential equations that allow us to generate these models. I don't have time to go through this in detail, but most important, the take-home message from this work is we try to combine mechanistic models that are interpretable because they are built on knowledge, so we know the components and we know networks, and that's one part. And then we try to couple that with machine learning that will help us to analyze the big data sets that can handle high number of genes, because the mechanistic models cannot work on all genes. That's just too computational intensive. So we have to simplify things there. So we try to merge these two to get the best of both worlds, and that's a team within CropXR is working really hard on that. Then, of course, the last slides that I want to show you are about this translation aspect, which is also a very serious endeavor. How can you use knowledge between plant species? And that's really a big question, of course. Comparative genomics is really very important there, that you know how orthologs are present between species. There's all kind of data already available on the plant connectome, for instance, how proteins are interacting, what has been described in the literature. So it's really about mining a lot of different resources, and their AI approaches are really important, of course. So within this translator, we call that the translator work package, we are making use of what is developed in the demonstrator, but we are also producing our own data on crops. So also again on seedling establishment, and we are now preparing for a big transcriptomic experiment in lettuce, potato, tomato and cauliflower to look at seedling establishment and have a time series in drought and elevated temperature. And then of course this comparative genomics is really important, and there also we are making good advances to understand orthologous relationships and how to learn how we should use that in our modeling approaches. So then the big question, of course, is how to translate finding to breeding. Well, this is the last slide. So with Let Us Know, I think we have been really working hard to develop lettuce into a very strong model system, and lettuce is part of the Asteraceae family, one of the largest families of plants, whereas also chrysanthemum and artichoke and chicory, etc. So it's also a model system for that group of plant species. And we have provided a large amount of new knowledge and also tools that breeding companies are using themselves. The hard thing when doing this type of research is that we don't really see what is being used inside the company, because that's of course too confidential. So that's kind of difficult to trace back, but when we talk to them and talk with the bioinformatics people, they are using our resources every day. Now that's clear. We are now also building a follow-up program focusing more on the translation to breeding. But I think it was a good success that we have established a very strong basis for lettuce. There's also all kind of spin-off projects that have been generated, and in many of the labs that have contributed to lettuce now, lettuce is now an organism that people continue to work on because it's such a nice system. Then for CropXR, we intend to deliver the methodologies, this modeling and hybrid modeling approaches to understand the complexity of plant resilience, that's one part, and to provide leads for the molecular players that can be used to breed crops of the future. So you should think of allelic variants that you need to bring together, candidate genes that you should improve. So it can guide genetic design, selection of parents, the combinations of alleles that you need to bring together. So we're also within the program, we are using cauliflower as a model also to implement some of our findings because their elevated temperature is also very strong influencer for crop resilience. So we hope that we can translate our findings within the program into a crop, but the companies will do that within their own breeding programs mostly. So with that, I think I would like to thank all my collaborators within Let Us Know and within CropXR and you for your attention, and I'm happy to take questions.
Thank you very much. And now it's time for you. Who is the first person interested in making questions? Juan.
Hello, thank you very much for your talk. I understand you generate vast amounts of data in your own experimental fields, and perhaps I missed the part of whether you validate your phenotype, your genotypes. Just you select the most resilient genotypes. Do you validate them in real-world scenarios and use this data to feed your own model?
Yeah, so within CropXR, we're doing that now in Arabidopsis first. So what I didn't tell you that also in this demonstrator work package, we're also using a diversity panel, in this case of Arabidopsis, that we have generated based on climatic conditions where these Arabidopsis accession have been originally identified. And we are also screening, or we have screened that set for responses under elevated temperature and drought. So we can refer back to that. So things that we find in our model, we can link that to polymorphisms in this diversity set. And then the next step really is to test out these variants under real life conditions indeed. We're not there yet, but also in working in the crops, we're really planning to do that. And for instance, also in the potato program, which is already running in CropXR, actually they have started at the other end. They are doing field trials at the moment. They have done now 2 years of field trials for drought and limited nitrogen availability. And they are this year doing an experiment with 200 genotypes. So to also get solid field data that we can then link to our models. And then, yeah, this should work back and forward, but this is a challenge also, indeed, yeah.
Thank you very much for a very, very interesting talk. Concerning crops, the program, I understood that it's a collaboration between the private sectors and the public sector, and the private sector is also putting money on the project.
Yeah.
Now, and it's a nice combination between basic science using Arabidopsis, and the idea is to translate it all the knowledge to crops. How do you convince the companies to invest in a project which you are going to work in Arabidopsis?
Yes. So for some companies it's easier than other, but I think here we stressed that it's about the methodology development. And we could make a strong case that we should do the development on the methodology on the plant species that for which there is most information available. So I think that that helped to convince them, yeah. And then from the start, we engaged in serious conversations. So also at the application phase, we were in very close contact with all these companies. Especially, we have different grades in CropXR. We have full parties that contribute a lot. They have also more rights, and we have contributing party. That's a second level. So we built a program with these full parties mostly, and they were really, I mean, they are all struggling with complex traits, yeah, and how to proceed in the future with this. And they also see that they need to integrate AI approaches in there. And now we had the chance to build a big program where the companies could benefit from working also more on the basic development of this. And then at the end, so you saw on the slide maybe 96 million euro program for 10 years. And the companies together, they bring in 15 million, yeah, and then divide it over 20 companies. They have access to a very large program with a serious investment, but it's a good buy-in, I would say. So they were joining, so that was good.
Yeah, but Jack Monod, he found the answer to your question. All what happens in E. coli also happens in the elephant. So probably this is the good way to convince the private companies to work on Arabidopsis. Who is the next one? Next question. No question? Okay.
Thank you for your presentation. It's very nice all this collaboration between different companies, but how are they open to share the knowledge between the different companies, or how are going to work together? Because this is the private industry, so this competition between them, how do?
Yeah, so they, and then I speak mostly from my experience in the Let Us Know program, where we had also very regular meetings. So they're always happy to provide input to the presentations of the PhD candidates, etc. And you get also most information if you talk with them one-on-one, when they're not with their competitors. So that's, I was telling yesterday also, in setting up the program, we go to 2 levels of talks. We start with levels with individual companies, so because then they say the most. And then in a later phase, we also have joint meetings. But yeah, they will never really tell their core business what is really strategically important, of course. But for instance, in the Let Us Know program, they did a second field trial because the tip burn was really important for them. They struggle with this because it's so complex. They cannot solve it on their own. So we had selected a number of genotypes that had interesting features in relation to tip burn, and then every company did a field trial. They collected data, brought the data back together, and shared it again. Yeah, so I thought that was really great that they were willing to do that. But again, it's then on a pre-competitive topic that is so complex that they like to collaborate. And once they have good leads, yeah, then you will not hear from them again. But that's a fact of life. And we then take the advantage to continue on finding out the scientifically interesting stuff on why this trait is giving this phenotype, or how is it regulated, etc. And they see if they can use it in their breeding programs. Yeah.
More questions?
Hi, I just have a question related to the funding. This project is funded by Dutch, by NWO, right?
Yeah.
Are you also in parallel scouting other European funding to leverage this project, despite that it's 10 years project?
Yes, so we are looking into actually we want to maintain CropXR alive after 10 years. So because we are building this virtual institute and we are looking for other funding opportunities. But we have to make sure that it fits because that makes it more difficult. We have a public-private partnership, so things cannot just be shared like that. So I first have to have the approval of the companies if to publish something or to share data. So that makes it a bit more difficult. We are now exploring how we can have projects next to CropXR so that there is a good interaction, and that we can use our CropXR affiliation. So we are exploring that at the moment also. So we are interested to team up with others and then see how we can establish these collaborations. Yeah.
More questions?
Because also in Europe there are now multiple larger initiatives. I know at least in Germany there is, in Denmark there is, maybe in France. I don't know about Spain, but it would be nice if the individual larger initiatives could also be linked to each other. And then if there's extra funding available for that, that would be great. Yeah.
More questions? I have one question. In your particular case, you are combining phenotyping with transcriptomics. And when proteomics will come up in this scenario? I mean, I think that high throughput proteomics is super well developed now, even competing with transcriptomics. And it is closer to the phenotype than transcriptomics. Have you evaluated that?
Yes, so in CropXR, in the C4 work package where we work on trade-offs, there is a strong focus on the proteomic side. We are looking at specific complexes that relate to trade-offs that are regulating growth versus immunity or growth versus some other resilience traits. And there we have at least biochemists within that work package that have also a core proteomics facility, so people have access to that. But it's not, I think within CropXR we are still focusing most on the transcriptomic, but we have proteomics, and then in another work package we have a metabolomics approach, and then it's about combining these within the program and how that also contributes to the modeling. Yes.
Yeah, and another question is that in this complex collaborative frame you have described, they are involved big companies, isn't it? And what about small spin-off companies from the Netherlands? Are they also invited to participate in these programs or not?
Yeah, so the difficulty is that companies to be part of this program, they have to contribute also financially, and then for small companies it's often very difficult to contribute financially. So that's a problem that we should discuss with our governmental organizations, that they cannot join in these projects because they don't have enough means to contribute in this way. So that we should find another way. And then maybe in phase 2 of our project, so the second 5 years, we have allocated budget to set up collaborations with small, medium enterprises. So that has been earmarked for that. And the idea is that once we have developed some technology here in the first 5 years, that we are then also working with other smaller companies. Yes.
Okay. No more questions? Not? Okay. If so, please give Guido an applause. Thank you very much.
Okay.
Thank you very much. Very
Thank you.
interesting indeed. So
I wanna dance by the water beneath the Mexican sky, drink some margaritas by a string of blue lights, listen to the mariachi play at midnight. Are you with me? Are you with me?